Thank you all for still being here in the last talk. We are having today the closing lecture now. And it's a great pleasure for me to introduce our last speaker and also thanking her for having been here around here all the time, even if most of the meeting was actually in Spanish, but she was here coming to the talks and everything. So introduce you to Dr. Mona Khan, who is now working at the Max Planck as a principal scientist, Max Planck in Frankfurt. Mona was born originally in Singapore, but she did her PhD in Sydney at the University of New South Wales. Then eventually she moved to New York to do a postdoc with Peter Mombaerts, who is sitting here in front as well, and has become her collaborator since now. She's at the Max Planck, still collaborating with Peter, where she works on olfaction and the olfactory system, particularly in the olfactory epithelium. And she will tell us today about a very interesting story they have been hunting down since the last few years about visualizing SARS-CoV-2 in diseased COVID-19 patients. So thank you very much, Mona, for being here, and all yours.
Muchas gracias, Flavio, for the introduction. It has been an absolute pleasure to be here at this meeting. Thank you to the organizers for this invitation. Really appreciate it. So I'm going to share with you our story on SARS-CoV-2, which culminated in two major papers, and I will summarize the salient findings here. But first, this work would not have been possible without the heroic efforts of our collaborators. First and foremost, the surgeons, spearheaded by Lara Van Gaven, with her team of ENT surgeons from the hospitals in Leuven, Brussels, and in Bruges. The pathologists, Arno van Stapel and Dietmar Thal. The virologists, Dirk Jokmans and Johan Netz. Johan Netz, the tall gentleman in the back, is the head of the Virology Institute, the Rega Institute in Leuven. And last but not least, of course, the team in Frankfurt, headed by Peter Mombaerts, who is the director of the Max Planck Neurogenetics in Frankfurt. So let's talk about SARS-CoV-2 virus, which is responsible for the COVID-19 and consequently the pandemic. Now, the pandemic seems like a distant memory, isn't it? But the SARS-CoV-2, the virus itself, is still here with us. SARS-CoV-2 is one of the human coronaviruses which appeared in December 2019. It is highly pathogenic and spread very quickly around the globe. As a result, the pandemic was declared by the WHO around March 2020. And I think some of us still remember where we were and what we were doing when the pandemic was announced, since it was such a singular event, probably in our lifetime. As of October this year, the reported COVID-19 death, the official number is 7.1 million worldwide, with United States on top of the list, followed by Brazil. Now, if you remember, Brazil had this nasty variant of concern, the Gamma variant, with high transmissibility, and the Gamma variant appeared in Brazil around November 2020. As for Uruguay, the official number of reported death is around 7,600 cases. Now, the cause of death is often associated with acute respiratory failure, but the most common symptom is the loss of sense of smell. And we refer to this as olfactory dysfunction. And it's because of this neurological manifestation that the olfactory projection here in yellow has been a prime suspect for providing the virus with a direct and short route to the brain. So our two papers centered around one question: is SARS-CoV-2 neurotropic? Meaning, can the virus infect neurons and other cell types in the nervous system? Now, I will give you upfront the short answer, and that is, after examining 115 COVID cases, we found absence of evidence for neuroinvasion. Now, the absence of evidence, we accept, does not equal the evidence of absence. And in science, it is not possible to prove a negative. However, we stand by our findings that the virus is not able to enter the brain. But we found something else, which I will share with you in the upcoming slides. But first, let's talk about the culprit. Now, the virus itself is relatively simple in structure. It consists of a spike, membrane, and the envelope. Few parts, but together these components form a highly efficient and formidable virus. Now, the SARS-CoV-2 belongs to the order Nidovirales, which is a positive sense, single-stranded RNA virus. What does positive sense mean? Think of it like your messenger RNA, meaning that it can be translated directly into proteins upon entry into the host cells, making this virus highly efficient. Now, inside the virion, the RNA genome is a single strand of 30 kilobase, and they are wrapped by nucleocapsid proteins. And the nucleocapsid is responsible for holding the RNA genome together. The spike protein, on the other hand, is key for making contact with the host cell receptor, in this case ACE2. And what it does is that it initiates infection. Once the viral RNA genome is released into the host cell, the life cycle of the SARS-CoV-2 begins. And we took advantage of one aspect of the life cycle, and that is the replication transcription stage, which takes place in complexes called RTC, or replication transcription complex, which has been translated by the viral RNA genome. These are non-structural proteins. So the RTC synthesizes a complementary strand of the RNA genome that can act as a template, so the virus can make many copies of itself. And it's against this complementary strand that we design RNAscope probes called the sense probes to detect virus that is actively replicating in our tissue samples. And another important thing to note is that these RTCs are found in specialized vesicles called double membrane vesicles, and the DMVs are actually modified structures of the endoplasmic reticulum of the host cell. So at the subcellular level, these replication transcription activities are found around the nucleus, which we refer to as perinuclear localization, which we demonstrate these in our findings. So all in all, we took on a virocentric viewpoint by designing as many RNAscope probes to look at several aspects of the virus in order to catch the virus in action in our tissue samples. So now back to the question, can the virus infect neurons? Now if you look at this illustration, there is only a few millimeters between the olfactory mucosa located in the roof of the nasal cavity and the olfactory bulb. So the question is, can the virus hitchhike or piggyback onto the olfactory nerve to gain access into the brain? So to address this question, the surgeons resected tissue samples from three main areas: the olfactory cleft mucosa, the olfactory bulb, and the frontal lobe. Now, they also included respiratory mucosa from the inferior and middle turbinates. Now, we cannot not study the respiratory mucosa. After all, the virus is responsible for acute respiratory syndrome. So this endoscopic endonasal surgical procedure takes place at the bedside of the patient soon after death, with the aim of keeping the post-mortem interval as short as possible. For instance, in cohort 1, the post-mortem interval, PMI, was kept at 70 minutes, which is probably a record in the literature. So these samples were sent in formalin pots to Frankfurt for processing and analysis. So cohort 1 consists of 70 cases of non-VOC, non-variants of concern, and the alpha variant. And remember, these samples were harvested during the pre-vaccination era. We only had one case, COVID number 68, in this cohort 1 that was vaccinated. Cohort 2 consists of 45 COVID cases of the Delta and the Omicron variants. Now, these are examples of tissue samples that have been processed but before embedding. So for analysis, we opted for RNAscope platform, which is ultra-sensitive with high signal-to-noise ratio for unambiguous detection of the viral RNA. And we coupled that with immunohistochemistry for co-detection of the viral RNA and its antigen by confocal microscopy. Now, it turns out that the respiratory mucosa was a useful source of positive controls, because the respiratory epithelium is highly likely to be infected. So we tested our RNAscope probes, and we tested many antibodies against nucleocapsid and spike antigens before we found the right one that works. So let's take a look at a few examples. Now, this is a respiratory epithelium of a patient infected with Omicron and died around 4.5 days after diagnosis. Now, this image is an example of an acute and highly infected case. So here we use nucleocapsid antibody, written in capitals, to detect the virus. The nucleocapsid immunoreactive signal in red clearly outlines the shape of the infected cells, sometimes facilitating in the identification of the cell type. In this case, we recognize them as ciliated respiratory cells. The RNAscope sense probe, the two sense probes. Remember, sense probe is to detect replicating viral RNA. So the two sense probes, the signal, they are in the form of puncta or dots, and they are present in nearly every cell. And this is what we mean by acute infection. Let's take a look at another example of an earlier case from cohort 1. Here again, the nucleocapsid immunoreactive signal clearly outlines the shape of the infected cells, whereas the replicating viral RNA signal from the ORF1ab sense probe is in the form of dots of green puncta. And they are located in a perinuclear manner. Remember, we talked about the double membrane vesicles around the nucleus, and this is what we are seeing. This is another case. Now, this one has a very good example of a perinuclear localization of a replicating viral RNA signal from the N-sense probe. Yes. Respiratory, yeah, the respiratory epithelium still, yes. So we also show in our paper that the target cell type for the respiratory epithelium is the FOXJ1 positive ciliated cells, here shown in the form of blue puncta. Now let's move on to the main part of the story, and that is the olfactory mucosa, right? We start with the olfactory mucosa. This is an illustration of a coronal plane of a nasal cavity. The blue line indicates the region where the olfactory cleft sample was resected. Now, the olfactory epithelium, the main cell type in the epithelium is the sustentacular cells, here shown in purple. And the main function of the sustentacular cell is to provide support for the olfactory neurons. And the olfactory neurons here are in yellow, they are bipolar, and they express olfactory receptor genes for the detection of odorant molecules for the sense of smell. The other two types of cells are the basal cells and the duct cells. Now, this is a human olfactory epithelium of an uninfected patch of an infected case. So we're looking at a patch of uninfected region. Here we use tubulin 3 antibody, a classic marker for neurons and axons. The tubulin 3 immunoreactive signal in green clearly outlines the shape of the neurons. The RNA-scope probe for UGT2A1, which is a gene that is highly expressed in the sustentacular cells. The probe is in the form of blue puncta, and they are located apically in the sustentacular cells. So now we are in the olfactory epithelium. Yeah. So I call these two cell types the dynamic duo because of their close relationship with each other. Now, if we take a look at an infected region of the same epithelium, but this time we will use ermine antibody, ERMN, which is a marker, another marker for sustentacular cells, mainly labeling the microvilli of the sustentacular cells. And here you can already see what looks like a clump of ghost cells devoid of ermine immunoreactive and UGT2A1 RNA signal. And indeed, these are the infected sustentacular cells visualized by the nucleocapsid probe. Now, in this case, the nucleocapsid RNA level in a single cell is so high that the individual punctum can no longer be discriminated, and they are merged and fill the entire cell, like an immunostaining. And for this type of pattern of staining, I only see them in cases with high viral load. Normally, RNA-scope probe takes on the form of dots, right? Puncta. Now, you can see here that the food processes of the sustentacular cells are wrapped around the basal cells, but not infecting the basal cells, which is contrary to some findings in the literature. Now, as I said up front, that we did not detect viral RNA signal in olfactory neurons. So let's take a look at an example to illustrate this point. So in this tissue section of an olfactory epithelium, a very long stretch of olfactory epithelium, I pooled 4 probes for 4 olfactory receptor genes to label as many neurons that express these genes. So the signal for these 4 probes is in the form of green puncta, and they take the form or shape of this doesn't work of a neuron, right, with a cell body and a dendrite. And in this case, in this example, the replicating viral RNA signal from the S-sense probe is in the form of red puncta, and they're located apically in a long stretch of infected sustentacular cells. And if you take a look at the high magnification of this box area, there is no commingling of the red and the green puncta. Now, this case is our most informative case of an elderly patient who died 4 days after diagnosis from our cohort 1. This is another example of infected sustentacular cells where the nucleocapsid probe outlines clearly the shape of the infected cells, and we use OMP probe to identify mature neurons. Now, I want to take a moment here to talk about the sustentacular cells. Now, over time, the substanticular cells have been given many roles, from structural to phagocytic, and mainly from the work of rodent experiments. Now, these roles are primarily to provide support for the functioning of the neurons, and I call the substanticular cells the unsung heroes of the olfactory epithelium. And because of this intimate relationship between the two cell types, we could not help but speculate if the substanticular cells are attacked by the virus and can no longer fulfill their roles. This in turn will affect the normal functioning of the olfactory neurons, which can then lead to olfactory dysfunction. In other words, during an infection, the olfactory neurons are affected without themselves getting infected. So the mechanism of acute and sudden loss of sense of smell during an infection is still unknown. We can only speculate, could the substanticular cells be responsible for this phenomenon, or could it take place at the level of olfactory projection? Now for that, we need to take a look at the axon fascicles in the lamina propria. But first, a bit about anatomical structures. As the axons enter the lamina propria, they are immediately wrapped by ensheathing cells, a type of glia unique to the olfactory projection. As the axons and the ensheathing cells come together, they form axon fascicles and become progressively bigger in diameter as they project to the olfactory bulb. Now, these axon fascicles, in turn, are wrapped by yet another unique cell type called the perineural olfactory nerve fibroblasts, here in magenta, a very long name for a cell type that is so poorly characterized. So back to the question, can we detect viral RNA in the axon fascicles? And the answer is no. Instead, we found them in the interstitium outside the axon fascicles. Let's take a look at an example. Now this was a patient infected with Delta variant and died 3.7 days after diagnosis. What you see here is a longitudinally sectioned axon fascicle, a very large axon fascicle, and we used two antibodies to identify these bundles. The first one is P75. Remember I mentioned that the perineural fibroblast is poorly characterized, but the one thing that is well established in the literature is the use of P75 neurotrophin receptor to specifically label these cells. The other cell type is S100 beta, a classic marker for olfactory and sheathing cells. Here you can probably already notice an array of nucleocapsid puncta in red in the interstitium outside the axon fascicle. And if we take a look at a high magnification of this region, we did not detect viral RNA signal in the axon fascicle itself. And here you can see very clearly the P75 immunoreactive signal clearly demarcating the outer layer, the outline of the axon bundle. This is another case, also a patient infected with the Delta variant, died 8.3 days after diagnosis. Here you see the nucleocapsid probe signal stays clear of the tubulin 3 immunoreactive axon fascicle. So another thing I want to mention here, if you take a look at this image, you probably could not help but wonder, surely the trailing viral RNA can hang on to the external surface of the axon fascicles and make their way to the olfactory bulb. So this is the next question. So as the axon fascicles penetrate the cribriform plate through the foramina, they are technically called the fila olfactoria. And as they enter the cranial cavity, The external surface of the fila olfactoria is contiguous with the leptomeninges. It's a tissue consisting of two types of tissues, the pia mater and the arachnoid tissue. And the leptomeninges actually covers snugly the olfactory bulb. So back to the question, can the viral RNA that we just saw in the interstitium outside the perineural fibroblast make it to the leptomeninges and subsequently into the olfactory bulb and the frontal lobe? Let's take a look. So let's take a look at the olfactory bulb first. This is probably our best olfactory bulb image, sagittally section, capturing both the bulb and its tract. We use SSTR2A antibody, somatostatin receptor type 2 antibody, to detect the leptomeningeal layer, which is in green. And we use tubulin 3 antibody for the neuronal component of the olfactory bulb. Now, after examining 115 olfactory bulb cases from COVID cases, we found absence of viral RNA signal anywhere in the parenchyma of the olfactory bulb. However, we found sporadic RNA signal in the leptomeninges as we suspected. So here the SSTR2A immunoreactive signal in green labels the leptomeningeal layer. So not only did we find sporadic viral RNA signal in the leptomeninges, we also found nucleocapsid immunoreactive signal in the leptomeninges. Now this piece of evidence tells us that these could be free virions that are circulating around the leptomeningeal layer. One of the most curious findings was actually the detection of viral RNA in the lumen of the blood vessel. Here identified by the endothelial cells is defined by the PCAM1 green puncta. Now it's very rare to detect viral RNA in the blood vessel. This was a case of a patient with an active oncological condition and died around 8 days after diagnosis. So from the olfactory bulb, now let's move on to the final part of the story, and that is the frontal lobe. So the frontal lobe tissues came from two regions, the gyrus rectus and the gyri orbitalis. So the question is, can we detect viral RNA signal in the frontal lobe? So you know the proverbial saying, looking for needle in the haystack? That's exactly what it felt like, hunting down for red dots or any kind of signal in the parenchyma. But luckily we didn't find them, so I think that is in a way good news. But like the olfactory bulb of COVID-19, we found them confined within the lumen of the blood vessels in some cases. Here we use CD31 antibody and PCAM1 RNA-scope probe to identify the endothelial cells. Now this was a very sad case, a patient with a malignant tumor, got COVID, died within 40 hours of infection, the shortest interval in our cohorts. So this patient died due to an increased intracranial pressure. So he died with COVID-19 and not from COVID-19. There is a distinction here. And most of the cases in our cohort were of patients who died from COVID-19. This is another example of a case. And an important point that we make in our findings is that the viral RNA presence in the lumen of the blood vessel does not extend beyond the glia limitans perivascularis. And the astrocytic end feet of the glia limitans perivascularis can be identified easily with aquaporin 4 antibody here shown in blue. So I will end our story with a collage of images that I didn't have time to show you, but they can be found in our two papers, and I will summarize the findings here. So at the respiratory mucosa level, we showed that the respiratory ciliated cells are the target cell type for the virus. And we demonstrate a perinuclear localization of the replicating viral RNA in acute cases. At the level of the olfactory mucosa, we found that the sustentacular cells are the target cell type for the virus, and we failed to find evidence of viral RNA in the olfactory neurons. Now, in the case of the olfactory projection, we found viral RNA present in the interstitium outside the perineural fibroblasts, and we proposed that these fibroblasts, perineural fibroblasts, can act as a barrier to protect the axon fascicles from pathogens and viruses as they safely project to the olfactory bulb. And finally, the final component of our story, we failed to find evidence of viral RNA in the parenchyma of olfactory bulb and the frontal lobe. But we found viral RNA presence instead in the leptomeningeal layer. And also, this viral RNA, when they are found in the lumen of the blood vessel, they are confined within the lumen of the blood vessel and does not extend beyond the glia limitans perivascularis. So with this, I would like to acknowledge the core members of the team from our lab. Sungjun Yu and Sumin Choi worked tirelessly throughout the lockdown and the pandemic. Marnik Klesters, MD PhD student from KU Leuven, who coordinated all the samples that came to Frankfurt, and one of the surgeons that's responsible for resecting the samples, and Laura Van Gaven, who courageously came up with this idea of resecting samples, harvesting samples at the bedside soon after death. And also to Peter Mombas, who is the brain behind the whole operation. He kept the project together and responsible for writing 2 highly enriched manuscripts covering several aspects from the virology, olfaction, to understanding of the cerebral vasculature. And we thank the Max Planck Society for their funding in this project.
Thank you very much, Mona. Wonderful talk. So we have time for questions. Matthias, you want to make your question? No? Okay.
Thank you very much. Very fantastic talk. Wonderful pictures with 4 colors. I love them. I just wanted to ask you, what is your prediction why you lost the smell? Because of the sustentacular cells, they get into the virus. Have you get into which of the function of these cells are failed? Perhaps the metabolic or the supporting or the tight shantion they have around? I don't know if you, okay.
Very good question. I mean, to really show the mechanism of the loss of sense of smell due to sustentacular cells, you would need an experimental model, an animal model, and that is quite lacking. But indeed, as I said, we could only speculate at this stage the mechanism of the loss of sense of smell. For instance, you remember I showed UGT2A1, which is a gene that is highly expressed in the sustentacular cells. And in one paper they did on rats, they found that this gene is responsible for odorant metabolization in terms of terminating the signal from the odorants. So it got me thinking that if, let's say, the sustentacular cells are affected, UGT2A1 no longer function and cannot terminate olfactory signal that is keep coming in, perhaps, as Peter pointed this out, there could be just white signal everywhere and the brain cannot make sense of the sense of smell. So indeed one would need to do an experiment. For example, you could do EOG, electro-olfactogram. insert electrode and check whether there is still any activities happening from the olfactory neurons. But at this moment, we do not have an experiment that can directly address the mechanism, experimental animal. Yeah.
Thank you very much.
Questions?
Here, in the third row, you show that the the signal for the virus is sort of in the wrapping of the nerve, right? Could it be? Well, I agree with you that it could be protection, right, from the wrapping, but also could it be a way of entering the jumping from cell to cell to cell using the envelope to, sorry, the well, whatever is that network of cells to push the virus inside? Has it seen in other nerve entries into the nervous system?
So that was our concern when we saw this viral RNA present, as you said, in the external surface of the axon fascicle. Is a way. I mean, these are actually axon fascicles going into holes in the base of the skull. That's why when we looked at any kind of viral signal in the parenchyma, and that is the most important thing, are the neurons in the olfactory bulb or the frontal lobe infected? But we did not find viral RNA in the parenchyma, but they are on the outside. So in some selected cases, for example, as I said, 115 COVID cases, only 23 cases we found with this leptomeningeal layer. So perhaps some susceptible patients would portray this kind of phenotype.
Any other question? So, just one more question.
Okay.
My question. So, why would neurons not be infected? Don't they have the receptor? So neurons, have you looked for that?
So yeah, our main concern, it is very simple, right? You lost the sense of smell. What does the olfactory neurons do? They detect the sense of smell. Ergo, olfactory neurons must be the ones that are affected by the virus, meaning that it's infected. The one clue that I got was ACE2 is not expressed in the olfactory neurons, and SARS-CoV-2 target is the ACE2 receptor. But as Peter mentioned, they could be induced during an attack, during an acute case like this. So there is always a worry that these olfactory neurons might be infected. We have to worry, though, about the mutation in the spike protein. That's why if you look through many papers from the virologists, they are actively studying mutations in the spike protein. And hopefully, they do not bind to a receptor that is expressed in the neurons. Yeah.
Thank you. Yes. Last question.
Okay, thank you. There is a conformational change in the epithelium or something? Because the olfactory, the paper of Grossmaite, a Nature paper, show that you have some, these cells, olfactory cells, are also mechanical sensor. And then there is another paper of Takeshi Imai that say that you need that mechanical input to codify odors. And then each odor creates some kind of phase change, and then there is some kind of phase code to decodify odors. And then if you have some, I imagine that if you have some kind of change in mechanical change there, perhaps you lost the mechanical part of the input. You know what I mean? No, the respiratory potential, the one that internalized to the brain.
So this is the problem, right? A lot of the work has been done in rodents, in mice. And we don't know what type of cells. It's still unknown, the human olfactory epithelium. We know only a few cell types. So to address your question about Takashi Imai and cyclic AMP signal, we did one experiment, which I really didn't have time to show you in our Cell paper, and that is spatial transcriptomics, whereby you can draw a region of interest, an area of interest, and you take uninfected region and infected region. And we asked, is there any differential expression in terms of all the genes that are typically expressed by the olfactory neurons, including receptor genes? Are they increased or decreased? Perhaps that could explain the reduction in expression of the olfactory receptor genes, thus the loss of sense of smell, and we found no changes. And this is whole transcriptome analysis of the two regions. Welcome.
Well, thank you very much, Mona.
Yeah.
So with this,
Pleasure.